what gets me excited about AI as someone who does kind of basic science, especially in biology, it is the idea of foundation modeling. It's the idea that you can have models instead of having very specialized models for specific tasks, right? So in biology, what was that? Like, you need really to have large models, you need a ton of data. And in biology, the type of data that you have is kind of, X-ray images, scans, things like that, or like pathology slides. So these are the things that like we started with early on, and then a few applications here and there. But beyond that, science is data starved, right? So we do not have the kind of scope of data that you need to train really large models to really take advantage of the emergent properties that these larger models really have. The promise of modern AI, and specifically foundation modeling, is this idea that you can build really large models on relatively cheap, kind of self-supervised data that forms a foundation for going into areas that are much more data starved, and with a lot less data, be able to fine-tune that model for a specific task, but now in a much smaller regime in terms of data set sizes that you need. Again, the idea being that these foundation models have pre-learned a lot of biology on their own by just interacting with large swaths of data, and so they have a much easier time getting to an answer as opposed to kind of starting with a very large model for a small data set that will never work. So I think that is fundamentally why I think this time is different from like other approaches to AI, because we have come to realize that instead of having different models for different tasks, we can have foundation models that then we can kind of retrain and gear towards different applications, including biology.
Welcome to Discovery Engines, a podcast on the people and the platforms who are accelerating scientific discovery. I'm your host, Nabil. This week, in partnership with NVIDIA, the nonprofit research organization, the ARC Institute, released Evo 2, the largest scale open source AI model for biology to date. Trained on over 9 trillion DNA tokens from over 100,000 species across the tree of life, Evo 2 is a generalist model that can, for example, predict whether a specific genetic mutation that we have might cause a specific disease. In this episode, I sit down with Hani Goodarzi, a core investigator at the ARC Institute who was part of the core team that built this model. As part of their work on this model, Hani and his team collaborated with Greg Brockman, co-founder of OpenAI, who helped their team tackle some of the massive engineering challenges that come with building and training a model at this scale. One of the applications of Evo 2 that I find most exciting is its ability to predict the pathogenicity of certain cancer-associated genetic mutations. Hani's got a unique perspective on this. A quantitative and computational biologist and an associate professor at UCSF, Hani co-founded Exai Bio, a startup focused on the early detection of cancer through cancer-emergent RNAs. In my conversation with Hani, we covered Hani's path into quantitative and computational biology, the emerging promise of foundation models, such as the ARC Institute's Evo 2, pivoting into AI for life science from an unrelated domain, and the importance of culture in absorbing these cross-functional team members, how ARC Institute's unique organization and funding structure enables innovative science, Hani's collaboration with NVIDIA and with Greg Brockman of OpenAI and what each learned from each party, the science behind Exai Bio's early cancer detection platform, UCSF's application of this technology for breast cancer detection, and Hani's view on why startups help bridge that valley of death between academia and industry scale-up. If you're interested in getting a sneak peek into the future of early cancer detection and treatment through the use of powerful AI foundation models, join us for this conversation. Oh, and one more thing. If you're enjoying this podcast, you can now support us by signing up for our free newsletter at discoveryengines.co. If you love this podcast and want to help us continue making new episodes, consider signing up for a paid subscription where you will get access to bonus content and research on the people and the platforms who are accelerating scientific discovery. Again, that's discoveryengines.co or discoveryengines.co. And with that, let's dive in. Hani Goodarzi, welcome to Discovery Engines.
Thanks for having me.
Absolutely.
Excited to be here.
Absolutely. So as I think about the way in which AI and ML has impacted us, especially over the last few years, the most obvious impacts that I think most people have felt have been in areas such as being able to generate text. If you're an engineer, being able to have a coding sort of co-pilot with you. We're moving into image and video generation. And as I think about the real promise of AI and the real way it's going to impact us, I think in the most significant way, it goes into areas that are these kind of deep scientific challenges that we haven't yet solved for. And one of the biggest ones that I can think of is cancer, which affects all of our lives. And so one reason I'm really excited to be chatting with you is that you were, as you said in our last chat, into AI and ML before it was cool. And not only that, but have been focusing that on the areas of cancer research through the work that you do as a associate professor at Department of Biophysics and Biochemistry at UCSF, a core investigator at the ARC Institute, which I'm excited to chat more about with you. And then through startups that you have co-founded, such as Vevo Therapeutics and Exai Bio, and that you're a scientific advisor to. So really excited to explore this wide space with you, and thank you for being here.
Absolutely. Looking forward to the chat.
Awesome. So let's start with just your path into science in the first place. So you have a Bachelor of Science in Biotech from the University of Tehran. You have a PhD in Quantitative and Computational Biology from Princeton. Just what got you interested in biotech?
Interesting question. The information that I have about kind of educational paths in Iran is like 20-something years old now. But the reality is that we come from a culture that is really into education and science overall, and so we do have in the backdrop of that, we have emergence of kind of career paths and educational paths that they kind of experiment with. And when I was looking at kind of options in college, there was this new program. It was a direct PhD program called Biotechnology, which was kind of a mix of a little bit of everything. So if you wanted to be a polymath, that was the way to go. And we will basically study different topics with folks who were kind of studying that as a major. Learn biology with biologists, learn microbiology from microbiologists, microbiology from microbiologists, but even beyond that, physics with physicists, engineering with engineers, and computer science with computer scientists. And so it was this kind of broad topic of how to bring engineering into kind of basic sciences, and specifically more so in biology. The reason that I started to think about, I really came into biology from the dry side, really, and what piqued my interest was really that topic of how kind of mathematics and computer science could be effective in biology. And one of the kind of key textbooks that a long time ago when I was in high school I came across was basically a book called Behavioral Ecology. And it basically dealt with like how behaviors have evolved in a lot of animals out there, and a lot of the book, interestingly enough, was focused on basically how evolution has solved like game theory problems, or like other mathematical optimization problems without actually sitting down and with a pen and paper and solving it, but actually solving it in practice through iterations. And so that was kind of the first time I started to think truly about biology as an intriguing and a complex field of research beyond kind of more memorization that we usually just associate with biology, or at least used to back in the day. And that combined with, at the time, I'm kind of blanking on the name, but I think there was a book called The Third Wave. It was talking about basically how human civilization has had jumped in productivity. I think agriculture was the first wave, industrialization was the second wave, and the author was, I think, was probably a book by Alvin Toffler, I think that's, if I'm not mistaken, about like biotechnology being like a third wave. And that was really formative for me. I think being a high school student back then, I really got excited about that, and that also played a big role in that. And finally, I also had mentors / friends who were really interested in biology, and their interest really piqued my interest. And that's how I actually got into kind of studying machine learning and neural networks for the first time. Was really through a mentor / friend, Dr. Arash Afraz, who's now actually, I think, at NIH. He's a PI at NIH. And he had just got into med school at the time. He was an alumni of our school. And he would take time to come and teach us things that he had learned. And he introduced us to this concept of artificial neural networks that he had just come across, and I got really interested in it, and dug a lot deeper to the extent that I started to using it in my own research as well.
So there was this I think sense from what I'm hearing in your story of being able to kind of scratch that itch as a polymath and be able to see how these different domains of engineering and biology are intersecting. Then there's the reality of actually doing, the academic work, seeing the state of neural networks back, in the day when we had access to, I imagine, a lot less compute and data. What did the reality of it feel like once you got into it?
Yeah, I mean, exactly what you said. I think ultimately, we went through and what we call an AI winter, right? I had learned and used neural networks in some of my research, but once I arrived at Princeton for grad school, folks had kind of moved on to, what was understood to be, a better approach to machine learning at the time, statistical learning, Bayesian models, things like that I retrained in basically as a graduate student. And again, Princeton was an amazing place, very strong computer science program that allowed us to kind of capture all of that knowledge. But at the same time, I did have the background that when kind of this modern AI took off again to really jump on board again very, very quickly. But the reality is that I actually gave an academic talk about kind of the idea of AI, modern AI, and why I think this time it is different. To be honest, we have gone along for the ride for decades around the promise of AI. And you gave some examples of why AI is interesting today and all these capabilities, but I think what gets me excited about Exai, about AI as someone who does kind of basic science, especially in biology, it is the idea of foundation modeling. It's the idea that you can have models instead of having very specialized models for specific tasks, right? So in biology, what was that? You need really to have large models, you need a ton of data. And in biology, the type of data that you have is kind of, X-ray images, scans, things like that, or pathology slides. So these are the things that we started with early on. And then a few applications here and there. But beyond that, science is data starved. Right? So we do not have the kind of scope of data that you need to train really large models to really take advantage of the emergent properties that these larger models really have. The promise of modern AI, and specifically foundation modeling, is this idea that you can build really large models on relatively cheap, kind of self-supervised data that forms a foundation for going into areas that are much more data starved and with a lot less data, be able to fine-tune that model for a specific task, but now in a much smaller regime in terms of data set sizes that you need. Again, the idea being that these foundation models have pre-learned a lot of biology on their own by just interacting with large swaths of data, and so they have a much easier time getting to an answer as opposed to kind of starting with a very large model for a small data set that will never work. So I think that is fundamentally why I think this time is different from like other approaches to AI, because we have come to realize that instead of having different models for different tasks, we can have foundation models that then we can kind of retrain and gear towards different applications, including biology.
Well, I know that you're doing some really innovative work in building these very large foundation models in various roles, one of which is at the Arc Institute. I want to get into that a little bit later in our conversation. Before I do, there was you had mentioned you were interested in working in the dry side of biology and research. At some point, you had this opportunity, I think, with Rockefeller in New York, where you, it sounded like in our last conversation, got your first taste of what real research into cancer looks like. I'm curious what inspired that, I would say, move on or into the wet side, and what struck you about that experience?
Yeah, good question. So, as you mentioned, I entered biology from the dry side, and first couple of years in graduate school, I was primarily doing dry research, and a lot of my work was actually focused on doing kind of looking at cancer gene expression data sets that had published at the time. So me entering grad school kind of coincided in an explosion with gene expression data sets that were emerging from different diseases, a lot of them in cancer. And so being a data scientist, that was kind of a natural progression of what we could do, is like building these kind of meta-analytical approaches to kind of harmonizing and integrating these various data sets. So that was really my introduction to cancer research by and large, not through a deep understanding of cancer as a disease, but really this happenstance of I'm a data scientist, I like to wrangle data, and there's a lot of data here. Of course, things evolved over time. At the same time, while I was on the dry side, I was really fortunate to be in a lab that actually did have a wet lab side. So I did my PhD with Saeed Tawazei, who at the time was at Princeton. Now he's at Columbia, and he himself had trained in Harvard, and he had moved over to Princeton to start his own lab. And Saeed had actually built a hybrid lab, which was very rare at the time. So there was actually a computational and experimental side to the lab. The experimental side was doing basically early days of pioneering research in functional genomics. At the time, of course, there was no CRISPR to do human functional genomics, so a lot of it was in bacterial genetics and bacterial genomics. But basically, Saeed's lab on the wet lab side was developing tools for kind of approaching systems biology of genomes and functionality of genomes. And so in the backdrop of my own research, I also had access to that training as well, just mostly through osmosis. And I also did pick up some skills on the wet lab side as well, around like systems biology, how to do microarrays, which are now extinct, but how to basically do systems biology in terms of some microbiology measurements. Now, going back to my work on cancer, once I published my work as a graduate student, having been a naive graduate student, I thought, this will matter a lot, right? That I think we had been able to point out, key vulnerabilities, key changes in different human cancers, and in some sense, giving identity to what it means for a cancer cell to be a cancer cell, and kind of what are the underlying cell biological programs that are driving that pathological process. And I expected that to, be not necessarily splashy, but like leave a mark, right? But in reality, I don't think that's how science works. So I don't think in reality anyone will really, at least at the time, pick up a computational paper and be like, oh, I'm going to spend the next 5 years of my thesis project on this interesting observation that a computational biologist has made. So I realized very quickly that if I truly believed in the power of computation to reveal insights about biology that will otherwise remain hidden, I needed to be able to speak that language and to actually prove that point. So that was really the impetus behind me thinking, once I graduated, there was kind of a fork in the road. Do I, continue down this data science, machine learning path, or do I take a step back and kind of rethink what the tools I need in my tool belt to be able to really be an effective scientist? At the time, it was very clear that biology is an experimental field. I think that gap has closed quite a bit, again, thanks to advances in modern technologies. But certainly at the time, the answer was that yes, absolutely, having access to know-how in terms of how to generate the kind of data that you need, but also how to validate your own insights to actually prove the point. Again, I was very fortunate to find the kind of mentors who were willing to kind of take me in as a computational biologist and retrain me, and which is, as you can imagine, it's not a quick process at all. I was not particularly good at it or anything like that. So I ended up doing a postdoc with Sohail Tawazi, who's Saeed's brother. So I studied in the family, but I met Sohail through our lab functions, and he actually helped me with some of my early work in Saeed's lab as well. So when he had a young lab himself at Rockefeller, and again, he was very kind and supportive to take on someone who had done very little cancer research, to take them on as a postdoc and basically retrain them in aspects of cancer biology. As you mentioned, how to do mouse experiments and grow tumors and engineer cell lines and all of that. So it was really in that period that I learned that language and I became capable of kind of taking my insights that fall out of computational packages and actually evaluate them and prove some of those insights in experimental models.
Well, I love how you almost fell into cancer research as a data scientist, where you were looking to solve, it sounds like, an intriguing problem, where there was data available, where you could really sort of use your skills. And then it led and opened up this new path for you. I'm curious, now I feel like there's an increasing number of what I hear of like tech bio startups and folks who may have been using AI ML for, in finance or completely unrelated fields to life science, who are being either actively recruited or co-founding companies in this sort of tech bio space, who are trying to solve these really, complex, systemic diseases that we're all, dealing with. I guess I imagine you must have an appreciation and empathy for the journey that those folks might be taking. Is that sort of, as you maybe look more broadly as how we accelerate and advance our research into this space, is that something that we should continue, or is it important for folks to start maybe more from a deeper biology background before applying the AI ML stuff?
I don't think there is a need for everyone to wear multidisciplinary hats. I think you can have a diverse team that comes from different backgrounds and are really experts in their own respective fields. But it's good to have team members and leadership who also have the know-how on both sides, who can serve as the glue and kind of translate or at least ease transfer of knowledge to the extent that is possible. But I think all of that is really only possible in a setting when you're dealing with folks who respect the other side, the other disciplines, and that is not as common as you would think. I think humans tend to be very biased in their knowledge and know-how, and an engineer thinks every problem is an engineering problem, and sometimes it is, and sometimes it isn't, and so the willingness to really understand that other disciplines are bringing something to the table will create an environment where a very cross-disciplinary team can actually work together to solve really big problems. So I think that the hardest problem to solve is actually exactly that. Like, how do you build multidisciplinary teams that function coherently, despite the fact that not everyone in that team is necessarily a multidisciplinary researcher. And I have done this repeatedly in my lab. My lab is a sizable lab, going from folks who are fully dry to fully wet. We almost never have fully dry or fully wet projects, so almost every project is a team project. And again, I try to serve as a glue that holds it all together, and not only translate and kind of shape, how one side can help the other side, but also help put in context how much effort each side really takes. So that's actually one part of underestimating what the other side brings to the table is often not understanding how much effort things really take, right? So having folks who are cross-disciplinary lets you tap into that and actually know, this analysis, how difficult was it actually? Like, how much credit should this person really get for having done this, right? Is it a click of a button, or is it actually some new way of doing this, right? So I think having folks in the team who can gauge that and let people know, like, what is the contributions here, will also go a long way. And ultimately, places like ARC are kind of masters of that. Like, they're built around this cross-disciplinary, multidisciplinary ideas. And in addition to my lab, as you mentioned, you named some of the companies that I've co-founded and I work with. And again, the same applies there as well. How do you bring a cross-disciplinary team into kind of working together to be the most effective versions of themselves?
That emphasis on culture is so interesting. And I, in a previous life, did a lot of work in helping organizations solve their kind of systemic problems using design thinking tools. And one kind of core concept of design thinking is being able to essentially walk a mile in somebody else's shoes. And so there would actually be these learning journeys or immersions that we would set up where somebody would actually have to do the task of somebody from a different functional department within an organization to get an appreciation for what it all entails. To be able then to problem solve more effectively. I imagine that gets a little bit more challenging in the technical domains that you're describing. And I think one way that you're solving for it, it sounds like, is yourself and others who have those kind of broader skill sets can serve as the glue for those teams.
Absolutely. I mean, ultimately, what is important to note is that I don't think it's news to anybody that multidisciplinary research is valuable. Right? And being a multidisciplinary scientist is very enabling. But where the rubber meets the road is that, well, it's difficult. It needs more training. It needs more effort. So you need to spend as much time on one and the other. And if the effort is not sustained enough and large enough, you kind of run the risk of being mediocre at multiple things, right? And that is not enabling at all. So, my advice to my own trainees and students and postdocs in the lab is exactly this. I think you have to pick sometimes, right? Just really the brutal honesty that being a multidisciplinary researcher requires a ton of sustained effort. We're not talking about just like a training period. For example, when I retrained as a cancer biologist, it took about 9 months. That's a long time, and you need to have access to a training environment that lets you take 9 months to just retrain. But even aside from that, then now that is just the beginning, right? Now, as a cancer biologist, I have to go to cancer conferences, read cancer papers, just be on the cutting edge of like where the cancer field is. But then as an RNA biologist, I need to do the same. And then as a AI scientist, I have to do the same. And interestingly enough, each of these were actually more manageable because cancer was a big field since I started. But both the RNA field and AI field were much smaller. And I think the progress was just a lot slower, if you will. But really, since the past few years, with the rise of COVID and the excitement around RNA vaccines, and kind of RNA blew up as a field, and so it's a lot more active as a field than AI. The story since 2015, 2016, just an explosion in how quick the progress has been. That even 6 months is like ancient, right, in AI time. So that when push comes to shove, you just really have to be willing to put in the time and effort that goes into being on top of multiple fields of research, and really to stay in touch with basically abreast of what is happening in each of these fields.
So you had mentioned ARC Institute as being one of the masters of cross-disciplinary work. So for folks who don't know about the ARC Institute, a couple of sort of quotes from the website. One is, it says, ARC's mission is to accelerate scientific progress, understand the root causes of diseases, and narrow the gap between discoveries and impact. It also has a really interesting organizational model, as you mentioned, with these 3 core concepts of core investigators, such as yourself, technology centers, and translational programs. So I'd love to go through those each in a minute. Before I do, though, could you share more about the founding vision of ARC?
Absolutely. I think the way that I personally think about ARC, and may or may not to some extent be divergent from how our founders think about ARC, so this is really my opinion, is that ARC is an experiment in science, right? So, and a lot of these institutes are, I think the underlying thought behind ARC is, how do we unblock scientists to do science and to do the best science that they can? And this is verbatim from some of our, what some of our founders say. So the way that the thought process, came to be is that during COVID, a few folks got together and started this granting process called Fast Grants. The goal of it was to really deploy capital for scientists to study COVID, and basically unblock as quickly as possible a lot of COVID research. And I think it exactly did that. But as part of that process, they also did a survey of academics who had received those grants, to basically ask some general questions and specific questions about the mechanism. I wasn't one of them, so I haven't seen the survey myself, but I've heard about it. And what came out of that survey was this interesting observation that a lot of scientists are saying that if they had access to funding that was not tied to specific projects, they would do things that are different from what they do. So basically, the current granting mechanisms are kind of funneling them into specific types of research that is not necessarily what they think should be the best use of their time as scientists. So that was interesting. And again, our founders, 2 out of 3 are scientists themselves, understanding that we are putting for a sizable lab, you will put in like a third, if not half of your time into just fundraising for that RAP and grant writing. So are you truly doing your best science if you're spending so much time talking about science and writing about science more so than actually doing it? So that was kind of the idea of core investigators comes out of that if we basically give core funding to scientists for a sustained period of time, a long period of time, and just let them do the best science, whatever that means to them, like what would the shape of that be? So that's kind of one side of ARC. The other side of ARC, again, going back to grants, the way that I think about it is that this kind of grants way of thinking also leads to every problem being or approached as like a quantized set of 4 to 5 years, where like 1 or 2 graduates and postdoc can work on this with another one, and that needs to be a unit, right? So, and then of course you can string them along over many years to get from A to B. That was your destination, but you have to kind of zig and zag towards that destination. And a lot of problems are simply not that small. I think the best example that I usually make myself is kind of what is the equivalent of the Large Hadron Collider of experimental physics in biology, right? So what are the scales of projects and problems that you need sustained effort across a large team, a cross-disciplinary team, with a lot of funding and a lot of support to be able to actually tackle, right? And again, there are a variety of approaches to address this. For example, NIH does consortia. Like Human Genome Project was one example of that. ENCODE project was another example. So NIH does hold this kind of team projects towards like solving big problems every now and then. And then there are other agencies like Chan Zuckerberg Institute, for example, has its own federated approach to taking on some of these big problems as well. And ARC came up with this idea of basically leveraging technology centers. So technology centers are a team of scientists or professional scientists who work together across 5 technology centers, each with their own expertise to tackle what we call ARC initiatives. So these are these type of big projects, big problems that ARC wants to tackle as an institute. And it was really founded on 2 initiatives. One is the Alzheimer's disease initiative, kind of how to model and study and ultimately, tackle Alzheimer's disease as a problem. And the other one is the virtual cell initiative, how to build an AI model of the cell. Again, like this happened many years ago. I mean, everyone these days talks about the virtual cell initiative, but and everyone have their own kind of definitions also of what they really mean by that. But ARC was founded on this idea many years ago and has a very clear definition and goal of like what it means to us as an institute to create a virtual cell model.
Well. I love this idea of using, in a way, the scientific method, not just to do science, but to rethink how science is done and do these organizational experiments. And I think it's interesting, you had mentioned that some of your initial work in cancer research was with the Rockefeller. You then mentioned Chan Zuckerberg Institute, which has been funded by, Mark Zuckerberg and his wife, and, some of the resources that they got from starting their own tech startup. Patrick Collison, I think, of Stripe, co-founder of Stripe, is one of the founding donors, as I understand, of the ARC Institute. So I think it's interesting how there's a new generation, essentially, of folks who are maybe taking their capital and resources and enabling scientists like yourselves to do experiments in how you're organized and how you're funded that weren't possible before.
Yeah, I mean, that's absolutely the case. I think, as you said, there are a number of tech founders out there who are trying to bring their own solutions to like how we should approach science. And in all honesty, I think we would need different types of solutions for different types of problems. So different organizations might find their own solutions to each of these problems, and I think actually power to that diversity, right? I think we should run these experiments and see what works and what doesn't. And the reality is that we are not really running a lot of them, right? The reality is that, again, much of biomedical research is funded by NIH, which again, in this uncertain climates, we are actually paying for that kind of monopoly of shaping science and what it really means in kind of biomedical applications. In some sense, I think ARC has already been successful as an organization that can recruit really smart scientists, that can build this kind of cross-disciplinary teams and can tackle some big problems that hopefully in the next few weeks you'll hear about. But I think another mode of kind of success for ARC, which I think we should hold ourselves as ARC Institute members to that is kind of replication, that other places start other ARCs and bring a similar model to other problems. I think that is really the ultimate form of success, that we can have like a diversity of solutions to the kind of how we approach science and specifically problems in biomedical research.
Well, in your relatively early days at ARC, right? It's a relatively young organization. you've had some pretty, I would say, significant announcements. So recently a partnership with NVIDIA was announced, where I think NVIDIA talked about ARC's biology and machine learning researchers are working with NVIDIA's engineers to scale the potential of AI models to design, predict, and understand biology. This partnership will build on ARC's previous work with the Evo model, which is capable of both prediction and design at the level of DNA and RNA and proteins in single-celled organisms. In parallel, I think one kind of fun story that caught my eye was, I follow the co-founder of OpenAI, Greg Brockman, on Twitter, and I noticed that when he took a sabbatical, he posted a picture as he was wrapping up that sabbatical. You were in that photo. He was at ARC, and he was describing how he had spent some time at ARC, and from his Twitter post, which is pretty lengthy and interesting, he says, overall, it makes it feel like the AI field has been creating a data abstractor over these past 80 years, which can be pointed to any kind of data and derive some useful sense of its structure to help make all of our lives better. He then says, there's a lot of exciting discoveries waiting to be had, coupling computational techniques with ARC's wet lab capabilities. So some really great partners in AI. Maybe just double clicking a bit on Greg's post there. He's coming in, as we maybe talked about earlier, right, of folks that have these, really amazing kind of AI, structural engineering type systems engineering backgrounds, combined with, the expertise that you have in science. Like, what do you feel that you learned from each other's perspectives in that partnership?
No, absolutely. I think exactly. I think it doubles back to what you mentioned earlier, that how do you actually build and take advantage of cross-disciplinary teams, and someone like Greg and the type of impact that they've had on large language models, and how can you leverage that for some of what we want to do in biology. The reality is that, as someone who has done machine learning and AI in academia for the most part, going to tens of GPUs is basically like the extent of what we have done. And it turns out going to thousands of GPUs is really, really hard. Who knew, right? Again, that's like some of those things that I touched on earlier, that, we don't really necessarily respect what we don't know, right? We don't know how much, anyone in my situation, might think that, well, we train something on 100 GPUs, now we do it on 1,000 GPUs. What could go wrong? It should go 1,000, 10 times faster. But in reality, that's not how things unfolded. And thankfully, Greg was there, right time, right place to really come and teach us how it's done, how we can actually approach this problem of distributed training and a scaling training, large language models in biology. And what I was really surprised by was also kind of how in the woods he was, that, he would actually spend time, fixing problems, and he would actually write these really long, debugging threads that was very educational, for all of us. So he was an amazing partner. I think we would not be here without really his support for the team. He also, to be honest, his star power was really crucial in helping even form the team. If you have a team that Greg is part of, everyone else wants to pay attention and work with this team in a variety of forms, which actually brings us to NVIDIA. We've been really great thought partners around kind of how can we not only build and train these kinds of really large language models that are useful in biology, but also serve them and make them kind of, provide them to the community in an easily accessible way. That, again, it's not something that we as academics are really good at, right? I think ultimately realizing that, we are researchers, and then we're not necessarily good at solving engineering problems on either side, right? And really being like ARC is a place where you acknowledge that, and at the same time in a position to actually reach out and actually ask for help and support from a variety of other organizations and folks who can actually help us basically fill in these gaps.
Well, we've been talking about kind of different ways of designing essentially organizations that do science, and your own path into scientific research and research into cancer. I want to get a little bit more into the science itself, and I'm going to ask you to bear with me as I reach back into my high school biology class here. And you had a great TED Talk. You said, "What if a simple blood test could detect cancer?" And you describe the journey, which seems to be very analogous to one of your startups called Exai Bio, about early detection of cancer when it's at its most treatable stage. And so as a refresher, DNA and RNA, nucleic acids made up of a phosphate group, a sugar, and a nitrogenous base. What makes RNA, in my understanding, kind of interesting is, the variations it has, such as, for example, mRNA or messenger RNA. So the ability to essentially leave like the nucleus of a cell and go out and pair up with some ribosomes and be able to create, for example, new proteins, which are so important to everything that happens in our bodies, from our bone structures to things like our insulin regulation and antibodies and so forth. And as I understand it, what you had found through your research is that there are what you called these cancer emergent RNAs, or I think you branded them oncRNAs, exactly, which are these essentially small pieces of RNA that get secreted into our blood that serve almost as barcodes for example, different types of tumors that or a type of tumor that might be in our body. They're incomplete barcodes, so there may be a hint of what that tumor is, but there needs to be some kind of filling in the gaps. And your technology platform is focused on kind of detecting those barcodes and then being able to then map them back to specific types of cancers.
Exactly.
How am I doing so far?
Amazing.
Yeah? All right.
Very
great.
impressive. Yes.
My AP bio teacher from high school will be very happy to hear that. So let's maybe, hearing it from the expert's sort of perspective, what first even got you interested in this specific, I would say, technology application for early cancer detection?
Yeah, I think there's this tired trope that I think science is less about eureka moments and more about oh, that's interesting, right? So the bottom line is we did not set out to solve a liquid biopsy problem. We did not set out to figure out a way to do cancer detection from blood. That was kind of a happy coincidence of the science that we were doing in the lab. Largely around this, cancer emergent RNAs that you mentioned, and we were interested in, during my work as a postdoc, I'd come across some of these RNAs, and it's just that has just stayed with me. That again, that's interesting. That there are RNA molecules that I only see in the context of cancer, but not in healthy cells. And when I started my lab, one of the we built our lab on multiple ideas, kind of risk managed, right? So some of the less risky, kind of we know this is going to work, bread and butter, and some ideas that we are going to take, give it a shot and see what we see. So this was certainly in that category of high risk, high reward kind of projects that we were thinking about at the time with my kind of founding postdoc, Dr. Lisa Fish. Lisa was a graduate student in Sohail's lab where I was a postdoc, so we actually started my lab together. So she gets a lot of credit for actually having helped me build the lab. And she was an RNA biologist, and we had worked together earlier on a lot of kind of how RNA and RNA binding proteins work in the context of cancer. So the first problem that we started to think through was how many of these cancer emergent RNAs are there even there? So it turned out a lot. So we systematically annotated over many years a lot of these cancer emergent RNAs, and we made interesting observations. One is that there's no shortage of them. There's tens of thousands of these RNAs, and more importantly, they are not appearing randomly. There is a logic to their appearance that is tied to the state of the cell, which means that a lung cancer cell will have different cancer emergent RNAs, which, as you said, we call oncRNAs. And interesting enough, onc is not cancer, it's orphan non-coding RNAs. Orphan in the sense that they come out of nowhere. And so these oncRNAs are tied to kind of the state that the cell resides in. Kind of like the same way that gene expression reflects on the state of the cell. And a lot of other high throughput measurements of the cell will also reflect that. But what is different about oncRNAs is that we started to think about them as kind of digital kind of barcodes of cell identity, right? Gene expression is quantitative. It goes up and down. You know, if I have more of this gene and less of that gene, then I'm a cancer cell or a lung cancer cell. Whereas in the case of oncRNAs, you can say if I see this oncRNA and not that oncRNA, I would be a lung cancer cell. But initially, we were really interested in how these cancer emergent RNAs may even play roles in tumor progression. They could actually evolve new functions in the context of cancer. And that was the basis of the project by and large. But quite serendipitously, one day we made the observation that some of these RNAs are actually secreted because there are small RNAs and cells do secrete small RNAs. And we have been actually interested in that process as well. And we had a project in the lab focused on understanding RNA secretion and kind of the underlying grammar of that in terms of which RNAs are secreted and which are not and why. And it turns out some of these oncRNAs tap into the same program to find their way out of the cell. And once we knew this, then we knew that there was a cancer detection application to look at there. And if I want to kind of maybe expand a little bit on that front, you know, you have a tiny tumor in the body, but it's not isolated, right? It's still connected to the rest of the body. And via blood goes to the tumor, and the tumor relies on it for its survival and growth. And like any other cell, it does release material into the tumor, into the blood. So in principle, if you can catch what the tumor is releasing in the blood, you should be able to tell that there's a tumor there. Now the catch is that, well, all the other cells in the body are also releasing material into the blood, so it's a needle in the haystack problem. So you need some sort of really cancer-specific signal to be able to latch onto. So the first time that we tried to solve this problem, for example, was with mutations. Like mutations in cancer, somatic mutations in cancer, are cancer-specific. So if you can find them in the blood, then it means that there's a tumor that's releasing them. It turned out it's a bit more complicated than that, and so mutations for early cancer detection were not enough of a kind of signal to noise ratio, but DNA methylation emerged kind of as kind of the right approach for reading a cancer-specific signal from the blood. So these are pieces of DNA that are differentially methylated only in the context of cancer, and then are released by cancer cells because cancer cells die and they release the DNA into the blood, and you can sequence the entirety of the DNA in the blood. Of course, there's DNA from the other cells, but you know exactly what patterns of methylation you're looking for, and if you find them, then it means that there's an underlying tumor that's releasing them.
Could you explain quickly what methylation means?
So methylation is basically, there is this gap between kind of DNA and RNA, kind of how is DNA regulated so that not all parts of DNA are turned into RNA, and specific parts are kind of transcribed and expressed in a specific cells. You know, the idea is that every cell in our body shares the same genetic blueprint, but we get, more than 200 cell types in our body. All of that is based on how every gene is expressed, where it's expressed, how much it's expressed. And so this is the type of regulation that sits on DNA that does that in every cell. And one way that that process is controlled is via marks on DNA itself. Which is this methylation marks. So methylation is a chemical group that sits on DNA. You can add it, you can remove it, and that can silence or activate a gene based on its proximity. But again, because a lot of dysregulations happens in the context of cancer, one of these dysregulations is emergence of these methylation patterns that are cancer specific, the same way that there are these oncRNAs, there's RNA species that are cancer emergent, the same that are mutations in DNA are cancer emergent. So all of these are kind of cancer emergent signals, but the question is how reliably can you actually detect them from the blood? And the challenge with DNA, on the other hand, as effective as it has been, is that they are just cancers release very little DNA into the blood. So it's just it's very good signal, but it's very faint signal. And so it tends to kind of skew towards later stage cancers for you being able to detect them. Now, in case of RNA, and specifically oncRNAs, cells release them actively. So it's not kind of a dead cancer cell event, it's a live cancer cell. Cells, even normal cells, are actively releasing RNA, small RNA, as I mentioned, into the blood. So kind of you can have like orders of magnitude more signal. oncRNAs allows you to focus that signal on cancer cells and be able to know if I see these oncRNAs in the blood, I know that there was an underlying tumor that has released them. And again, we have built these patterns of oncRNA presence and absence that we are basically looking for in the blood.
Well, one of the, I think it sounds like early applications of this technology was around a collaboration with UCSF. I think I saw on the website about 200 breast cancer patients were part of this maybe study where you were looking to detect residual disease and help motivate additional treatment, whether it's required or monitoring. What specifically, I guess, was there a specific characteristic of breast cancer versus other types of cancer that made it more amenable to your technology? How did you land on that sort of important
really good question. The short answer is no. I think my lab studies breast cancer for the most part. We are studying breast cancer progression and metastasis. So that's where we are most comfortable. I'm part of the breast oncology program at UCSF. Very great mentors at UCSF, Drs. Laura Ventvier and Laura Esserman, who are co-directors of our breast oncology program, who also run this amazing adaptive, really the largest adaptive trial that I know in breast cancer, really any cancer for that matter, which is called I-SPY 2. I-SPY, which is now, I think, I-SPY 2 and like beyond 2 at this point. So I-SPY is a very valuable resource and it looks at localized advanced breast cancer and how in neoadjuvant context, so basically when patients are diagnosed, what kind of treatments are best suited for every patient, kind of this idea of personalized treatment to maximize response. And they have also, as part of this trial, looked at kind of different liquid biopsy applications for detecting minimal residual disease, including some of the DNA-based modalities. And because they were aware of my work, I pitched them this idea that we could actually take advantage of oncRNAs with small volumes of blood, what was really left at that time for some of these patients, to be able to actually see if there could be clinical utility, and that's what we showed. And this was actually partly before even Exai was spun out of the lab, but actually formed the foundation of that spin out. But Exai itself had a multi-cancer kind of approach. They've had some papers already around lung cancer and soon other cancers as well. But breast cancer remains very near and dear to our heart, not only because we are breast cancer scientists, but also the fact that breast cancer is actually one of the indications where they shed very little DNA into the blood. So for DNA-based modalities, they have been a very tough problem to solve, especially since for women with dense breasts, for example, where mammography sensitivity really comes down, there is a gap that kind of liquid biopsy can fill to really change detection, the kind of landscape of detection for these patients.
Well, the work that you're doing with Exai is one of 2 startups that you have co-founded. You're also a scientific advisor on a number of others. I'm curious about, as you make sense of your own entrepreneurial journey, what inspired you to take that path, and what purpose, or I say, what benefit do startups like those serve in this larger ecosystem of scientific discovery?
Really good question. I would say the way that I see the role of scientists, especially academic researchers, I think we are really good at solving 0 to 1 problems, right? Where you're like, I have this idea, let me try it. It becomes exponentially more difficult to go beyond there because, everyone has a small teams of dedicated to this specific problems, right? To give you some context, for example, like Exai was built on this long RNA work that was done, with the team of like 2-3 people in my lab, which then became a company 10 times that size of scientists, so dedicated to this problem that 3 people were solving. So that is kind of a given that we are really good at that early kind of de-risking of ideas. And a lab like mine where we do translational research and we have 25 people. In practice, there is kind of a portfolio of 50 or so projects, right? And if you put your mind to it, like every single one of them could potentially be a startup, but that's not how I think about the startups. I think a lot of things have to go right to think about, like, is it really truly worth it to take this 0 to 1 idea? I think, of course, others are more than welcome to come and take the one and make it 1,000. But for me personally, given what I do and my day job, does it make sense to take this 0 to 1, make it 1 to 10, so that it will actually be a spun out. Now, that gap, in some sense, industry is really good at the 10 to 100. Mm-hmm. There's usually a 1 to 10 gap that remains, and I think that's kind of the valley of death for a lot of good science, which is unfortunate. I think when the market is strong and when money is flowing, if you will, that gap closes, right? I think, to be honest, in the 2021 kind of market, it was probably even behind some of the scientific labs, and there was no gap, right? You could get an idea and run with it, right? But when the market is tough, like it is now, then that gap widens, right? Which again, not a good thing, because you're going to lose a lot of innovation. A lot of good ideas will just sit on shelves and not going to be actually applied to anything. So the question for me as an academic researcher is fundamentally which of these ideas are worth pursuing beyond kind of my lab and beyond my work as a researcher. Interestingly enough, only some of it has to do with science. The other part of it actually has to do with the team. And finally, kind of the market. Like, can I find the right people to actually come and take this idea? Because, I have a day job. I'm happy to support them, but to the extent that I can as a scientist. But in reality, it's a full-time team who is actually going to work on creating this company and actually drive it to success. I need to be able to trust that they can do it. And, my vision is kind of safe with them. That is really hard, like finding the right team that you think they are good custodians of like what you have spent maybe 5 to 10 years of your life on. And also that there is actually a real application for it, that this team that you have assembled is not going to just waste their time and build a company to nowhere. So all of these things have to really go in the right direction and really have to line up to really make sense to start a company. As you mentioned, we have done this a couple of times, and each of them I'm very excited about, both the science that has gone into it and then the companies that have been built on top of them.
Well, thank you for that perspective. Wrapping up here, so one of the things that I just love about our conversation today and about your journey is how playful and experimental you are in advancing science, whether it's through your own work from starting as a data scientist, moving into cancer research, to being part of organizations like ARC that are really an experiment, as you said, to trying your hand at successfully starting a couple of companies and getting venture capital and so forth as another yet approach and model to unlocking some of these challenges. For this podcast, one question I like to ask every one of our guests is, we've got an audience here of folks with talent, with access to resources, maybe know about policy and so forth. How could folks who are listening to this podcast support your work?
Well, my old website had a donate button. I'm just kidding. So I think the reality is that we do actually work with a fair bit of kind of volunteers. Like Greg was an example of that. So some folks do have the knowledge and know-how that can actually contribute to the work directly, right? But also there are folks who, if not directly, indirectly can interact with scientists. I think science communication and evangelizing science, I think, is something that scientists are not necessarily good at, right? And maybe even bad at, to be honest. I think there is a gap between, again, what we do on a day-to-day basis in terms of the research and science that we do, probably even today. Like how good am I at kind of simplifying ideas to a lay language that I can really communicate it to the folks, to really make them understand that this is valuable, that I'm doing it for a reason. I think a lot of folks can really get together and help evangelize science. And really also understanding that scientific method goes beyond just science. That scientific method is basically a process out of which truth really comes out. And we should be applying the scientific method to all aspects of our lives, be it policymaking, be it politics, be it engineering, whatever we are doing. That process of experimentation and data collection, analysis, thinking, and hypothesis generation, and validation, and kind of iterating through that circle applies anywhere. It's not just basic science. Of course, that's where it was first conceived and continues to be applied. But in practice, scientific method is more generalizable as an approach that we should be able to really apply it anywhere. And I think anyone and everyone should communicate that to kind of in all aspects of their lives and careers.
Well, Hani, as a polymath who was initially interested in applying engineering into new domains, to a scientist who's applying the scientific method not just to the research, but to the way in which science happens, I'm so inspired by your story and so thankful that you spent this time and shared it with us today.
Thank you for having me. It was a lot of fun.